Good morning, everybody. Here we are with our next lecture, that is third lecture for you, and today I am going to talk about the second part of your unit first from paper 6, that is water microbiology. Now before going to the topic, the basic you should know that what is water microbiology. So simply we can define is that study of microorganisms. Okay, so microorganisms which include virus, bacteria, algae, protozoa, and many other microscopic fungi. So study of microorganisms and their activities in fresh, estuarine and marine water, including spring, lakes, rivers, seas, which are the inhabitant of this natural water. So study of microorganism, their activity in the water is known as water microbiology. Now your first part from water microbiology is introduction. So the major water introduction is that water, it plays an important role in living system. In living system, both in case of animal, plants and human being. It is also known as universal solvent. Why it is called as universal solvent? Because each and every life, it depends upon the water. Now green plants and algae, they also use the water as an electron and hydrogen donor in photosynthesis. Many enzymatic activities are also dependent on the water. Of our total earth surface, 3/4 of our earth's surface is covered by water, hence it acts as an important habitat for microorganisms. So this is the basic introduction regarding the water. Now there are many diseases which are also transmitted by the water, human diseases. Mostly 80% of the infectious disease, they are transmitted through the contaminated water. Now why it is called as contaminated water? Because some pathogenic organism may present in the water and contaminate that water or make it unsuitable for drinking purpose. Next, we come to the point, that is sources of microorganisms in water. That means how the microorganisms are added into the water. Soil, air, sewage, organic waste, dead plants, animal, etc. Therefore, water contains all types of microorganisms. So, as we know, these are number of sources from which the microorganisms are added into water. So, water will contain different types of water. Some different types of microorganisms. Some coming from soil, some from sewage, some from organic waste, or some from various excreta. But all the organisms, they do not survive into the water, because there may be some water which may be or which may create unfavorable condition, and thus it doesn't support the growth and and they immediately die. Means some microorganism, they enter into the water, but because of the unfavorable condition of water, they do not survive. Now other sources, rain, it comes from atmosphere. Okay, so the atmosphere, it contains high amount of dust, as we have studied in air microbiology, that in the atmosphere there is a presence of dust, it is along with the microorganism. So when the rain, it falls from the atmosphere, comes in the contact with the microorganism present into the dust. Next, when rain, it touches the earth, so again the microorganism present on the earth, it adds into that rainwater, and thus that water get contaminated. So when it touches to the earth, it gets contaminated by the microorganism from the soil. Now initially, when the rain is slow or it is mild, the load of microorganism is high, but as the rain, it is heavy, now the water, it get dilutes and the microbial load, it get decrease. मीन्स काय, जेव्हा पाणी जेव्हा पाऊस इनिशियली कमी असतो, त्यावेळी त्याच्यामध्ये मायक्रोबियल लोड कसा असतो, जास्त, आणि जसं हेवी रेनफॉल वाढत जाईल तसं ते वॉटर काय होत जातं, डायल्यूट होत जातं. आणि वॉटर डायल्यूट होत गेलं की त्याच्यामधला जो मायक्रोबियल लोड आहे तो पण कसा होतो, डिक्रीज होत जातो. The microbial load in the stored water bodies may be decreased due to sedimentation, exposure to sunlight, increase in temperature. As we know, आपण घरी पण बघा खूप वेळ जर पाणी ठेवलं तर त्याच्यामधलं जे काही डस्ट पार्टिकल आहे ते काय होतात, खाली बॉटमला सेटल होतात. दॅट प्रोसिजर इज कॉल्ड ॲज sedimentation. Similarly, the microbial load in the water supplies or water bodies which get reduced by the sedimentation, by exposure to sunlight, or by the increased temperature. Next, all these reasons, these are the natural reasons why the water get contaminated by the microorganisms. Next is, apart from these natural sources, water also gets contaminated by improper treated sewage. Now treated sewage is, if the sewage is not treated properly and if it added into the water, the organism present in the sewage, so maximum or varieties of organism they come from the sewage into the water. Now industrial effluent are added into the water. Okay, next reason is runoff from the agricultural and animal waste. Runoff means what? When the excess of water is flowed from the agriculture, the organism from the soil or the animal waste, it comes into contact with the water. There are some water bodies which are used for washing the clothes, for bathing, or some animals are also washed into that water. Such water gets contaminated by such clothes, that is dirty clothes, or the organism present on the body of animals, human being comes into that pond. Similarly, swimming pool water also get contaminated by the skin infection. For example, some pathogenic organisms they are present on the skin. If such persons are utilizing the swimming pool, then the organisms from their body are also added into the swimming pools. So these are the purposefully contaminated reason. Okay, and earlier we have seen about the natural reason the water gets contaminated. Okay, so this was the sources of water. So some natural sources, some man-made sources or purposefully made contamination sources of microorganism into the water. So next point is the fecal pollution of water. Okay, water is adequate quality and quantity is essential for healthy life. Okay, so as we know, water it should be of good quality and also the quantity of water should be more. Okay, there is a close relationship between the water and human wealth, because why? If water get contaminated, human health internally get affected. Okay, so why? Because many diseases are associated with contaminated water. Common risk to health is due to the fecal pollution of water. Okay, so due to the contaminated water, many diseases are occurred. And now what is mean by fecal pollution? When the pollution of water supplies, okay, it may be river, it may be ponds, when it is added by a fecal matter of a human and animal, then it is termed as fecal pollution. What is fecal pollution? Pollution of water supplies by addition of fecal matter of human being or animals, then it is termed as a fecal pollution. And as it is contaminated by the fecal matter of human and animals, it is purposefully the addition of pathogen, because there are number of pathogen present in the intestine of human being and the animals. If such pathogens are added into the water bodies, then that water bodies get contaminated. Pathogens are always associated with the fecal matter of human and other animals. Therefore, such pathogens enter into water bodies due to the fecal pollution of water. So, it is the fecal pollution of water. It is one of the severe case in the contamination problem. There are certain bacterial species which are mostly causing the fecal pollution, especially the coliforms. Coliforms like E. coli and related organisms. Now you have studied in your practical about the E. coli. Okay, so it is called as other bacteria, fecal streptococci. Next is Clostridium perfringens are the normal inhabitant of the large intestine. Therefore, these bacteria are constantly present in the feces. So, these three bacteria and many more other species are present in the feces. Presence of any of above this bacterial species in water acts as an indication for the fecal pollution in water. Means above this organism, from this organism, if any organism is present in water, then that water is called as fecally polluted water. These organisms also indicate the presence of intestinal pathogen. Therefore, in the routine bacteriological analysis of water, testing for indicators of fecal pollution or fecal coliform is done. Now, why it is called as indicators? Because whenever you take a sample, for example, you are taking a river sample, it is very difficult to find each and every pathogen present into the water. So, and determine whether it is contaminated or not. So, if we identify one of the indicator organism in that water, then we can tell that that water is contaminated by fecal pollution. म्हणजे जे काय वॉटर सॅम्पल आपण आणणार आहे त्यामधला एखादा एक ऑर्गॅनिझम जो त्याच्यामध्ये प्रेझेंट आहे तो प्रेझेंट असेल तर सगळं वॉटर फिकली कंटॅमिनेटेड आहे असं आपण सांगू शकतो. तर तेच ट्रीटमेंट आपण पुढं यूज करायचं म्हणून इथे दिलं आहे. देअरफॉर रूटीन बॅक्टेरियोलॉजिकल अनालिसिस ऑफ वॉटर testing for indicator fecal pollution or fecal coliform is done. Okay, so this was about the fecal pollution. Now, which organism or who acts as the indicators for fecal pollution? It is ideal to look for individual specific pathogen in the water. So I have told you earlier की एखादा इंडिव्हिज्युअल पॅथोजन शोधणं त्या पाण्यामध्ये थोडंसं डिफिकल्ट आहे, पण तेच आपण काय करू शकतो, एखादा इंडिकेटर त्या पाण्यामध्ये आपण शोधू शकतो. सेम इट इज आयडियल टू लुक फॉर इंडिव्हिज्युअल स्पेसिफिक पॅथोजन इन वॉटर, बट द नीड फॉर द इंडिकेटर्स ऑफ फिकल पॉल्युशन इज नेसेसरी. मीन्स प्रत्येक पॅथोजन आपण फाइंड आउट करू शकतो, पण स्पेसिफिक इंडिकेटरच का शोधायचं, तर त्याच्यासाठी काही लिमिटेशन्स दिलेत. काय लिमिटेशन्स आहेत, का इंडिकेटरच फक्त शोधायचं? बिकॉज पॅथोजन एंटर्स इन टू वॉटर बॉडीज ओकेशनली ऑर व्हेरिएबली अँड डू नॉट सर्वाईव्ह फॉर अ लॉंग पिरियड. म्हणजे जर आपण तो पर्टिक्युलर सिंगल ऑर्गॅनिझम न शोधता प्रत्येक पॅथोजन शोधत गेलो तर ते डिफिकल्ट का आहे, कारण वॉटर बॉडीजमध्ये जे काय पॅथोजन असतात ते खूप वेळसाठी सर्वाईव्ह होऊ शकत नाही. आणि आपल्याला जो पॅथोजन शोधायचा आहे तो पॅथोजन, तो सॅम्पल आपण लॅबोरेटरीमध्ये नेऊपर्यंत तो पॅथोजन तिथं राहीलच असं नाही, तो तिथं पॅथोजन काय होऊ शकतो, तिथून एस्केप होऊ शकतो. म्हणून इथं दिलं आहे बघा, देअरफॉर दे कुड बी मिस्ड इन अ सॅम्पल सबमिटेड टू द लॅबोरेटरी फॉर ॲनालिसिस. इफ देअर नंबर इज व्हेरी स्मॉल दे मे एस्केप फ्रॉम द डिटेक्शन प्रोसिजर इन द लॅबोरेटरी. म्हणजे आपल्याला जो पॅथोजन आपल्याला शोधायचा आहे तो पर्टिक्युलरली तो लॅबोरेटरी जाऊपर्यंत तिथं मिस होऊ शकतो किंवा त्याचा नंबर कमी असल्यामुळं तो त्याच्यामध्ये डिटेक्ट होऊ शकणार नाही. तर हे लॅबोरेटरी प्रोसिजर जे आहेत दे आर ऑल्सो लेंदी. सो लॅबोरेटरी प्रोसिजर आर लेंदी अँड मेनी पीपल मे कन्झ्युम वॉटर अँड एक्सपोज टू द इन्फेक्शन बिफोर डिटेक्शन ऑफ पॅथोजन. म्हणजे स्पेसिफिक पॅथोजन आपण आयडेंटिफाय करत बसलो तर काय होऊ शकतं की लॅबोरेटरी प्रोसिजर लेंदी असल्यामुळं ते आयडेंटिफाय होण्याआधीच तिथले वॉटर सॅम्पल पीपल काय करू शकतात, कन्झ्युम करू शकतात अँड दे मे लीड टू द इन्फेक्शन. सो हे एवढे सगळे लिमिटेशन्स जे आहेत, एक लिमिटेशन काय सांगितलं की पॅथोजन जास्त वेळेसाठी तिथं राहत नाही किंवा तो मिस होऊ शकतो किंवा त्या डिटेक्शनमध्ये येऊ शकत नाही आणि लॅबोरेटरी प्रोसिजर लेंदी असल्यामुळं पीपल कन्झ्युम करतील आधी आणि मे कॉज द इन्फेक्शन. सो बिकॉज ऑफ दिस लिमिटेशन्स associated with the pathogens, techniques have been developed for demonstration of known bacterial species. म्हणजे आपण अशी टेक्निक यूज केलं आहे ज्याच्यामध्ये आपण काय डिमॉन्स्ट्रेट करणार आहे, नोन बॅक्टेरियल स्पेसिस. म्हणजे आपल्याला माहीत आहे की इकोलाचं कॅरेक्टर माहीत आहे, त्याचे बायोकेमिकल्स माहीत आहे, तो पर्टिक्युलरली तोच ऑर्गॅनिझम त्याच्यामध्ये प्रेझेंट आहे की नाही ते आयडेंटिफाय करायचं. इफ सच ऑर्गॅनिझम इज प्रेझेंट then we can tell that water is contaminated by fecal and many pathogens may be present. See, techniques have been developed for demonstration of known bacterial species of fecal origin, particularly organism of coliform group. म्हणजे coliform ग्रुपमध्ये येणारे जे का ऑर्गॅनिझम्स आहेत पर्टिक्युलरली तोच ऑर्गॅनिझम आपल्याला माहीत आहे आणि तोच त्याच्यामध्ये प्रेझेंट आहे की नाही बघायचं. आणि जर तो प्रेझेंट असेल तर वॉटर नक्की काय झालेलं आहे, फिकली कंटॅमिनेटेड झालेलं आहे. ह्याच्यामध्ये प्रत्येक पॅथोजन डिफरंट पॅथोजन आपल्याला आयडेंटिफाय किंवा आयसोलेट करायची गरज नाही. नेक्स्ट, now we have studied that there may be coliform. We have to find the indicator organism from the group of coliform. Now what is the coliform? You know this because you have studied this in your practical, so we will take it. Coliform, what are they? These are the organism which includes both aerobic and facultative anaerobic group of organism. Character is they are gram negative, non-sporulating bacilli, and they produces acid gas from the fermentation of lactose at 37 degrees Celsius within 24 to 48 hours. The most common group of coliform is the Escherichia coli. So this is very important. So Escherichia coli is an indicator organism of the fecal pollution. Now what is the advantages of testing the indicator of fecal pollution? Coliform, particularly E. coli, they are constantly present in both healthy and diseased human intestine in large number. We know that in diseased person also the E. coli is present, and as well as in the healthy person the E. coli are present. Now during billions of this organism, that is E. coli, may be excreted daily in average of the person. So whenever the water is contaminated, there is 100 or maximum percentage of chances that there is presence of E. coli, which can be used as an indicator. This organism, it lives longer in the aquatic environment than that of the human intestine. Now why other reason we find the E. coli? We have studied in earlier point that pathogen do not survive for a longer period of time. Okay, but this E. coli, it survive in the aquatic environment for longer period of time than in the human intestine, and it can be detected within the short time. So presence of this organism can be detected within the short time. So thus presence of E. coli in water is a warning signal that the water is subjected to dangerous pollution, thus important indicator of fecal pollution. Okay, so E. coli, it indicates that water is being subjected to pollution. Now there are three group of bacteria which are invariably present in the human and animal feces. Now which are the bacteria which are invariably present in the human feces are fecal streptococci, in that Streptococcus faecalis. Next is Clostridium perfringens, Streptococcus species of anaerobic, that is lactobacilli species. Now out of this Streptococcus, it is fecal, it shows the fecal pollution in both water and food. See, especially it is used as a fecal pollution for both water and food. But we can't tell that exactly which of these three is involved in the fecal pollution, because none of these or other coliform is a perfect indicator of human pollution, because all these occur in soil, in animal dung, as well as the human excreta. Means we can't tell that out of these three which is responsible for human pollution. Okay, so this was our last point. Okay, so we have in this lecture we have studied about your first point in water microbiology, that is introduction to water microbiology, which are the sources of microbiology, what is fecal pollution, and what is the indicator for the fecal pollution, and what are the coliforms. So this was your first lecture in water microbiology. Now after, in your next lecture, we are going to study in detail about various techniques used to determine the presence of coliform into the water and many other points. Okay, thank you.